Morning class. So today is in zoology 7th microbiology subject. We are going to see the very first topic is about the history of microbiology, or you can say how the microbiology come as a discipline in our subject that you are going to study these days. So how the foundation of microbiology has been built. So there have been many discoveries or foundations that led this microbiology to come in existence. So here we have to see what kind of discoveries and who the microbiologist or the scientist who had played a major role in making the foundation of the microbiology. So let's begin with the introduction part. So basically, if we talk about like what is microbiology, so in the very simplest form of definition, we can say that microbiology is a study of microorganisms, microbes, or you can say the very tiny apparatus which cannot be seen by your naked eyes, or for seeing them, you needed a microscope view. So anything which we can be able to see under the microscope, we can call them the microbes or microorganisms. So basically studying these microbes or microorganisms, their study is called the microbiology. So these microbiology, this is a very diverse group of living organism basically, or you can say the study of these living organisms, which we do under the microbiology. They have a very diverse, huge era or domain or kingdom, you can say. In this, they are going to include the algae, archaea, bacteria, cyanobacteria, fungi, protozoa, viruses. So basically, these all the things which I just mentioned or they're written here, the microorganism, they all are very tiny apparatus and you cannot be seeing them clearly by your naked eyes. You can see them in colonies, but you cannot see their individual structure. For identifying their individual structure, you are going to need some microscopic help. So here, if we talk about their, you can say their importance to our environment. So basically, most of the microorganisms they are harmless. If we see, if most of the microorganisms, if they are not, or you can say if they are toxic or poison, our life on Earth cannot be possible. So because of their 99% of their goodness, can be able to live on this earth because our existence today's existence so belong to them, or you can say we are basically dependent on each other. It's like in an ecology where things have to be related to each other with their for their survival. So that's the point here, that if the microorganisms are not present around us, so there will be a very complicated situation we have to face, because lots of the functions or lots of the process in the environment they are going to happen just because of these microorganisms. So if we talk about their 99% are good, only 1% are bad, or you can say the bad in the sense that they can make cause disease, they can have some pathogenicity, or you can say sometimes they are more lethal. So these 1% they can be bad for our health or our environment, but other than these 1%, the 99%, they are the good microorganism and they help us to our survival. So this is about the microbiology, like what is microbiology, why we need to study the microbiology. So if we wanted to see how or what kind of good bacteria is around us, how they're affecting us, and what kind of bad bacteria and what they are doing to our environment or us. So all this study is come under the microbiology. Now, if we talk about their historical approaches, so if we why we are studying the history, actually history is a very important. Like some people think why we need to know like who discovered it, who founded it, until and unless you don't understand the struggle what you seeing today, you are never going to see their real value. Okay, so basically for examination of the real value what you have today, you needed to know like what kind of efforts to be given for this present situation. So that's why, or sometime when you study this historical reviews, or when you see what pattern have to be followed for some discoveries, then the new ideas generated in your mind. Like when you thinking something and you feel like this is so stupid, how this thing can be possible in some uh, great things. So at that time, when you are going to study the historical part, there was the scientists like you and me that who are going to do research, and they are also have these stupid ideas, but they after all not stupid in the today's world, because they are thinking future. So that's why we have to teach you history, or you can say the foundations or discoveries. Then only you can go up with the discover mind, like what kind of the things or thought can come into your mind when you talk about the discoveries, or when you think about anything new. Something whenever the thing is new, it's feel like that this cannot be happen, or this may be not right. But believe me, these things, if you have something, some base or some provens, you can prove a new theory by them. So now, in this microbiological history, this whole discovery part, that was divided by into the four major eras. So that you can say the four major eras is like first the discovery era, at the point we found about the microorganism. Like someone clicks, or in the some scientist their mind and clicks that yeah, there can be something which cannot be seen, but they are present, like this kind of thing. With when they the very first idea of having microorganism around us, that was come in the discovery era. After that, the transition era. So the transition era is all about the brainstorming thing, like is the talk or whatever we found it in the discovery era, is it right or not, or some people going to prove that points which have been come in the discovery era, or some going to neglect them. So basically, when we are proving something or we are disproving something, we have to make some theory against that. So that's why, for approving something and disapproving something, you needed some clues, or you needed some proofs. So that's why in transition era, that the all thing is going on. Like some was going to support the discovery era, and some was going to disapprove that. So for approving and disapproving, they work a lot. Now the third come to the golden era, and golden era is all about the proofs, like the all things which come, or all the discoveries with the proof has been introduced in the golden era. So you can say a very true way of existence of microorganism and their belonging to world is come into existence in under the golden era. After this golden era, the modern era, in which we are living in today's world, and we are having a lot of discoveries onto the new bacteria, their protein synthesis, their functioning, drug discovery, medical microbiology, and a very broad aspect of using all the techniques which we had been discovered before, and now we are mutating them, we are variating them. So these are the things which we are going to do in the modern era. Now let's talk about the very first era, that is called the discovery era. So this discovery era, basically, that was you can say that was called like the spontaneous generation. So, if we see what is the difference between a spontaneous and something which having by some cause. Okay, so spontaneous is the anything which generate naturally. Okay, we do not have to do anything artificially into them. So that's why this has been called spontaneous generation. Why we are calling this spontaneous generation, we will see by the Aristotle comment. Like Aristotle in 384 to 322, that is beyond time, very like you can say very earliest of the timing. So and other believe that the living organism could develop from the non-living material. So this was his thought, but he was, he was thinking. He was, and this is a very important thing. Like thinking is a very important thing. If you cannot think, you cannot make anything. So, like Aristotle, maybe you're right or wrong. That is the second thing, but the thing that is the first thing, and that is important. So if you're wrong, then you are giving a world that this cannot be happen. But if you're right, then you are giving the world a new thing. So that's why Aristotle did that thing. Maybe it is right or not, but you can say, he fit himself into the pages of history by commenting something. So others believe that the living organism could develop from the non-living material. He think like that. Yeah, and at that time, or the time he was living, is like 384 to 322. At that time, any person can think like that because science was not developed that time. So that's why this idea came into his mind. After so many long time later, in 13th century, Bacon described the disease caused by a minute seed or germ. So in the 13th century, someone felt like the disease which we are having inside our body, they cannot be spontaneous. Okay, like they may be caused by something. Okay, so something is the reason that we are having disease, we feeling sick, or anything bad happening to the environment. There must be a cause. It cannot happen spontaneously. But the Aristotle thing, that was totally spontaneous. But Rogan Bacon have a little sense about like the thing which they cannot be spontaneous. Now they come into the picture Antony von Leeuwenhoek, the very important man of the history. 1632 to 1723, he was a legend. Why he was a legend? Because he was the first man, you can say he was the father of bacteriology and protozoology. Because with some proof, he finally have things on the microorganism. So basically, he gave the description of protozoa, basic type of bacteria, yeast, and alga. So you can say these are the very broad spectra of the microbiology, and he discovered them. So that's why for this major and huge discovery of his, we call him the father of bacteriology and protozoology. Also, this man, this von Leeuwenhoek, he developed the microscope. You can see a very primitive kind of microscope just for the enhancing something. So, and this was a major reason why Anthony von Leeuwenhoek able to see these microorganisms just by the discovery of microscope. And this discovery that was also done by the Leeuwenhoek, and because instruments are very important. Like we have things, we know that this can be happen, but if we don't have instrument, we cannot do that things. So like the people like the Rogan Bacon, he know that something is present surrounding us, like to causing these things, but he don't have anything to prove that. But Leeuwenhoek, he has something to prove his theory. So that's why he describe, or you can say, make something which can prove your theory. So that's why Leeuwenhoek make the microscope and prove his theory. In 1676, he observed and described microorganisms such as bacteria, protozoa as amaniculus. So basically, this aliquus, that suffix, is used when we are talking about some tiny things like molecules. Okay, so he didn't coin the term microbe or microorganisms. Okay, he described them as an animalculus. So like animals are the big thing, but they are very tiny things, so he cannot call them perfectly animals because they are not perfectly animals. So that's why he called him something like a small version of the animals, so he called them the animalculus. So the term microbe is used by Sadilot in 1878, but the term that we are using in today's world, microbes, that was given by the Sadilot, just after 100 years after the 1723, 1878, he gave this term. But animalculus, the primitive, or you can say the initiative term for the microorganism given by Leeuwenhoek. Now, that was all about the discovery era. If we see the transition era here, so what happened inside the transition era? So the very first, Francesco retired. So this transition era, that was all about the brainstorming thing, but some very important, you can say, discovery has been come from this transition era. First was the red eye. He showed that the maggots would not rise from decaying meat when it covered. So he thought, like, if I cover these meats, decaying meat, so maybe these maggots, this thing will not going to be present on my meat. And when it did, he didn't get the maggot. So basically, what he think, if the covering of the thing basically inhibiting the microbes to come on the surface and causes them the disease or causing any kind of harm. So this was a good thing. We also using that today. We covered the things after the use. Now the John Needham in 1713 to 1781, what he supported the spontaneous generation theory. He's still on that, like that everything can come naturally. But his reasons for this, why he was thinking like that, why he was thinking the spontaneous generation theory is true, because he did the same thing what was proposed by the red eye. Did I propose that if you're going to cover something, microorganism cannot do the attack? But he did the same thing, but still the microorganisms are present and their thing get basically spontaneously microbes can come on their mutton. So when he see this, he believed that the whatever the red eye was said that was wrong, because he did that and he is getting a new result. So basically research is all about that. Okay, when we are founding something, that is a search, but repeating that search is called research. Okay, and research is very important, because most of the time the people have to the scientists basically they have this ego problem. I don't wanted to call it an ego problem. Basically they have this thing in mind, maybe it is right or wrong, maybe the scientist who gave this, maybe he did not do his study proper. So they doing this research, and this is very healthy process for anything, for science development, from the environment development. This is a very healthy process. So that's why he proposed a tiny organism, and see that if he is going to cover it, still they are inside them. So that's why they can be present spontaneously. They can come from anywhere. They are the natural grower. They cannot be caused by something, or they cannot be stopped by something. So that's why this John Adam, he had his own reasons to support the spontaneous generation theory. After these whole, you can say, discoveries, Lazaro Spallanzani, he was, I personally think he was a good researcher of the time of transition era. What he demonstrated, that the air carried germs to the culture medium. This was a very fantastic kind of discovery, because in today's world we know that the air has several kind of microorganisms in that, and this thing clicks in his mind, and he proved that. Okay, he showed that the boil broth would not give rise to the microscopic form of life. The second very important thing he gave to us, like in today's world also, what we are doing, we are boiling things, we are making them hot to basically destroy the microorganism inside the thing. So that's why this thing was come from by Lazaro. So that's why I personally think Lazaro did the two very most future emanating thing about like this, that he demonstrated that the air carried germs, and second, he showed that the boiled broth would not give rise to the microscopic form of life. So these were the two very important discovery of the transition era. Now the come in the picture is the golden era. So basically, golden era, as the name suggests, something very good going to be happen this era, and the something very good, right? Or the golden man of this era was the Louis Pasteur. So Louis Pasteur is most of the people, or most of us, we are studying Louis Pasteur from our class like sixth or seventh, when we first very initiated the process of vaccination, or you can say so not only the vaccination, for the pasteurization process of the Louis Pasteur, because heating up the milk, and we know if we heat up the milk for a particular temperature to the specific time, then we can store it for a longer time. So this whole process of like pasteurization process that was given by the Louis Pasteur. And because of their modern support, or you can say their noble support, they has been called as a medical father of medical microbiology, or you can say in today's world we call him the father of medical microbiology. So Pasteur in 1897 suggested that the mild heating and 62.8 degrees Celsius for 30 minutes rather than boiling was enough to destroy the undesirable organism without ruining the taste of the product. What happened when we boiled something for a very high temperature or for the very long time? So at that time, most of the like some things, or you can say nutrients inside the our product, they can also be destroyed along with the products. So this thing cannot be very good for developing any kind of, you can say, the mutual funding in that case. So this purpose for not wasting out the product taste, Pasteur, what he did that basically you can destroy the microorganism, but along with destroying the microorganism, you can, what you can do, you can making the taste as it was before. So this process, actually the process was Lazaro, he gave that the thing like the culture medium or microorganism can be destroyed by the boiling, but the Pasteur suggested that the pasteurization process is better as comparison to the boiling process. So he invented the process of pasteurization, fermentation, and development of effective vaccine. So these were the three very noble things which have been done by the Louis Pasteur. The very first thing, pasteurization, that we just studied, fermentation, when we do the alcohol production, fermentation, and they develop the effective vaccines. So the vaccination process that was very new to that generation, and most of the people was feared that they cannot have vaccine because it maybe that vaccine make them diseaseful or they can be dead by that. So that was a very tough time for the pasture to convince people that it was good for health. Like in today's world, as we are taking the corona vaccines, oh, several other vaccines, we know that the vaccines are very essential and very needy for the living. So but in that time when the pasture need that vaccine, that was very difficult for him to convince people to take that. But still, he made it out and he made the vaccine for the rabies and anthrax. So the Pasteur demonstrated disease of the silkworm was due to a protozoan parasite. Another thing that Pasteur did, that at that time, when the time of Pasteur, like he was in 1897, right? So at that time, there was a famous silk route for the transportation, and I'm not telling you that the silk route just for the silk transportation, but what happened, the initiation of that route via the transportation of silk has been initiated, or you can say the very first thing which has been transported, that was silk. So that's why we call them the silk route. So that silk worm formation, or in that time, European country, they are fond for the silk. So basically, for industrial purpose, silkworm, you can say, for the conserving silkworm was their essential requirement. So silkworm was due to these silkworm, they are having some disease because of the protozoan parasite. So the major cause of their disease that has not been understandable by the industrial mentors or the scientist who was working. But the Louise was the one who introduced that the silkworm, they are not getting disease itself. They are having a cause for getting the disease inside them. And that is called the protozoan parasite. Now, what was the contribution of Louis Pasteur if we see the point wise? So, the very important, he coined the term microbiology. So, the subject you are starting today, microbiology, that was given by our famous Louis Pasteur. Second, aerobic and anaerobic. So, these two terms, aerobic and anaerobic, they are also very important because these terms were coined by the fermentation process, like this fermentation. So, if we talk about in fermentation, this aerobic and anaerobic condition, they are very important for understanding that. He disapproved the theory of spontaneous germination. That's is obvious. Nothing can be very spontaneous. Everything have their own causes to be germinate. He demonstrated that anthrax was caused by bacteria and also produced a vaccine for the disease. The anthrax disease, which was happening inside the livings at that time, so that was produced by a virus. He demonstrated that. So there is no spontaneous germination. He demonstrated the cause of that particular disease, and also he made the live vaccine. So he developed live attenuated vaccine for the disease. So, like in today's world, when we made the vaccine, we did not do the live thing. We take our dead RNA part and make the vaccine. But in that case, in time, the science was not so developed. But still, he make out a live attenuated vaccine for the disease, and it was working at that time actually, because maybe he knew some mutations in the living bacteria for not causing the disease, but just introducing inside the body. Now the come in the picture is John Tyndall. So the John Tyndall, 1820 to 1893. So what he discovered the cause of these microorganisms, or you can say, till now we study the microorganism do this, that, cause this, but he developed their initiating step, initiative. Like the, if we talk about a bird to be formed, the egg come first. Like don't get the hoof come first, chicken or egg, but egg is a primitive thing we for the making the chicken. So he discovered highly resistant bacterial structure later known as endospore. So this discover highly resistant means which cannot easily killed. So this resistant bacterial structure that was developed by the tendon, that was called the endoscope. So, prolonged boiling or intermittent heat was necessary to kill these spores to make the infusion completely sterilized, and this process is known as tyndallization. So, as he discovered this, so he named it after himself, tyndallization. So, what happened in the tyndallization process? We have to give a persistent heat. Okay, a time of heat is way more than the normal pasteurization. So the more heat and for the prolonged boiling, both the things along at the same time, they can be able to kill the these spore for a, you can see, for the complete sterilization. So that was the thing which was given by the John Tyndall, and this whole process is come into the fame by the Tyndallization. Now, Lord Joseph Lester, again simultaneously same time thing, like 1827 to 1912, he's the father of antiseptic surgery. So we know that this surgery, when we do the surgery or when we do the stitches, or if we have something wound, open wound, they can be separated by the microorganism, bad microorganism present in the air. So this concept, like the microorganism can cause a septic inside the body, that was covered by the Lord Joseph Lister. He discovered this. So he is the father of antiseptic surgery. Lister concluded that the wound's infection too were due to microorganisms. So the whatever the infection caused onto the screen inside the body of wound which are expanding, so they can be caused by the microorganisms. So that's why he devised a method to destroy microorganism in the operation theater by spraying a fine mist of carboxylic acid into the air. So he said, if we are going to operate something, so air, surrounding air should be clear and out of the bad microorganism or any kind of microorganism, because as he used the carbolic acid into the air, that he doesn't mean just to destroy the bad one, also the good one along will be that. So you can say that all the microorganism will be dead. So this whole process was given by the Lord Joseph Lister. So that's why we are calling them the father of antiseptic surgeries. Now, new one. come and picture Robert Koch. He was also very good for finding the bacteria can cause disease. So he demonstrated the role of bacteria in causing disease, like he preferred the technique of isolating bacteria in pure culture. So he was the very first who cultured the bacteria. Okay, so he taken out the bacteria from some place and he cultured them out. For culturing that bacteria, he used gelatin. But gelatin was not as good for culturing media. Why? Because gelatin is a proteinous thing, and proteinous can be digested. So protein basically digested by many bacteria. So if you are culturing the bacteria on the gelatin, so what happened? Bacteria will going to digest that particular protein. So there will be no protein and your culture can be destroyed. And also the gelatin have a very you can say the if it it's a melting point is very low, it's like the 25 degrees celsius. So if something have this less of the melting point, so always we needed to be refrigerated. So that gelatin was not a very good kind of culture media that was used by Robert Koch. But the Robert Koch demonstrated that the disease caused by the bacteria and we can culture out these bacteria for further purpose. Like Koch first relates, what he did, he isolated some microorganisms from the dead sheep. He cultured them out here and cultured microorganism he injected into a healthy mouse. So what happened to the healthy mouse? After some time that healthy mouse got died. So basically, he proven his point. He proven that these microorganisms can cause disease, and that disease can be this lethal for making a mouse dead. And you can say the most of the genetic pole of the mouse has to match with the humans. So if that thing can be act on the mouse, that the thing can be act on humans. So that's why postulates is all about that microorganism can cause disease and they can be lethal for destroying that particular organism. Now, the fan Alishmus has. In 1850 to 1934, he was a Bush assistant and proposed the use of agar in culture media as he was seeing that the Koch was using gelatin. So he had something in mind, and also like we can use something else to make it better for a culture media. So like what he used the agar in culture media. So it was not attacked by the most of the bacteria. So most of the bacteria, even in today's world, we are what we are using for the culture making. We using the gelling agent as a agar. So it was not attacked by the most of the bacteria. Agar is better than gelatin because of its higher melting point, and also their melting point is very high. It's like 96 point degrees Celsius, and solidifying is 40 to 45, just half. So that's why these properties are very good for culturing the bacteria because they cannot be digested. Their melting point is high, and also their solidifying is half on the temperature. So that's why these are the basically identical situation for culturing the bacteria. Now the Richard Petrie, he was a very important person because he developed the dishes in which we can culture the things. So he developed the Petri dish plate, which we are using today as a Petri plate, a container used for solid culture media. So basically, this is a container, this is a box which is going to contain our culture media. So in today also in our labs, we are using the Petri plates for doing the culture. Edward Jenner. He was the first to prevent smallpox. So the smallpox was very famous at that 18th century. So that's why he basically find it the cure and small. He discovered the technique for vaccination, and also the great Jenner was is famous for the you can say the very efficient kind of vaccination that was developed by the Edward Jenner. Alexander Fleming, he discovered the penicillin from Penicillium notatum that destroys several pathogenic bacteria. As there are 99% are good bacteria, but still there are 1% pathogenic bacteria. So Alexander Fleming what he discovered a very important antibiotic, one thing, that is penicillin. So this penicillin that he was discovered is for destroying the several pathogenic bacteria. Paul Ehrlich, 1920, he discovered the treatment of syphilis by using arsenic. So he did that. The treatment of syphilis, syphilis is a very common disease. So he treated that by using the arsenic. He studied toxins and antitoxins in quantitative terms and laid foundation for biological standardization. Also, what he studied, he like every bacteria, they have some toxins and they have some antitoxins. So, in their quantitative nature, so like what amount of the toxin and what amount of the antitoxins has been present in a certain kind of a bacteria, that was discovered by the Paul Ehrlich. Now, they were the all important scientists, scientists or microbiologists of the era. Now, here's some important discoveries. Like if we talk about the discoveries about the bats or bacteria, so the Hansen in 1874, that was a leprospecies bacillus, Neisser in the 1873, gonococcus. So basically, these scientists are responsible for the discovering these special kind of bacteria. Ogestrone, Staphylococcus, very important lab bacteria. Lawfare, Diphtheria bacillus, Rosix and Yersinia diphtheria toxins. So they are the about the bacteria. Now if we see the virus part, benzenic. Oh, that was very important and play an important role because he coined the term virus for the filterable infectious agent. So basically, what happened when the Benzenic identified the virus, because he made a filter for the bacteria which was causing the disease. So when he made that filter, but still some the thing, any other thing which passed that filter and caused the disease. So there was striking in his mind, like he already used a filter, how and some very small thing can pass that filter and cause the disease. And that time he discovered the virus. Pasteur developed rabies vaccine, that was important. Good pasture, cultivation of virus on chick embryo. He did when the Benzenic in 1898 he discovered the term virus. Then that good pasture, he's a not uh, the relative, he was some other pasture, cultivated the virus on the chick embryo. Charles Chamberlain, one of the Pasteur associate, constructed a prolactin bacterial filter. So this procalatin bacterial filter is basically filtering the bacteria out of any culture. Toad and Henry. They were also important because they found a very important kind of virus, bacteriophage. Bacteriophage are the virus which can eat the bacteria. So they were the very important discovery. Edward Jenner for the vaccination of smallpox. So these were the virus-related discoveries, and other one was the bacterial-related discoveries. Now, in the modern era or the era which we are living in, so here we can see the EOS virus. In 1901, the Nobel laureate is the one Behring. What is his contribution? Diaphragm antidotes. So he made that. In 1902, the malaria was identified by the Ronald Rose. That was very important kind of foundation. 1905, Robert Koch, he discovered about the TB. That is also important. 1908, mention of phagocytosis of the bacteria, the major nutritional mode. 1945, Fleming, penicillin. He was famous for that antibiotic. 1964, Watson Crick, structure of DNA. I think you all know them, Watson and Crick. They made the structural DNA. 1968, Holly Rana for the genetic code. So the genetic code was given by the Holy and Khuranas. 1997, Prashner for the prions for the making anything in a recombinant and genetic engineering. 2002, Berner and Hodges for genetic regulation of organ development and cell death. So, how the gene are responsible for the developmental and regulation process of a cell and how they are responsible for the cell death. So these all the discoveries was happened inside the modern era, and these are very important. And so on, there are a lot of discoveries which is going every day in a new form. And man, as we are developing with the science. So we have numerous things to study. So hope you are doing something, contributing the science for their developmental process. Okay, so these are some references if you wanted to learn more about microbiology and their existence in their life. So you can go through these references also. Okay, that's all for today. Thank you.